household was pretty stable. So one type of abuse may not show that kind of impact. Maybe you had sexual assault by someone in your neighborhood or a pastor or someone else that you interact with, but it didn't reflect on the family. But once you start seeing data showing 2 or more types of abuse in a family environment, my sense is that's a marker for the overall family environment. And probably what it's really speaking to is what kind of parenting and care did you receive, not just what kind of abuse did you experience, especially early in life. And it will have set the stage for your attachment. It will talk about those critical developmental periods that we discussed so much yesterday, because this is the environment in which you were raised 0 to 5, right? And this is the environment in which you experience adolescence. And so I think it probably doesn't matter so much what the types are. I think we can look at that, and we have, and some of our studies have shown different effects of neglect and abuse, or physical abuse can have very focal, seems to affect startle response in particular, which makes a lot of sense because it's sort of a very physical reaction, whereas emotional abuse we've seen has an effect on response to happy faces, which makes sense like we discussed yesterday. But overarching, this tells us about the family environment. And this is another way of slicing that same data, and it talks about age. So over here we have the age at which they first experienced physical abuse, okay? And then here, so either 0 to 5, 6 to 10, or 11 to 18. And then this is outcome for 3 different so depression scores, emotional dysregulation scores, and post-traumatic stress disorder scores. And these are all standardized because they're not on the same scale. But you'll see them here expressed in terms of standard scores. And what we see, I think, was notable given what we learned yesterday, which is that we see most of the impact for early life exposure, and then again for adolescence exposure. It's not that there's not impact in middle childhood, but there's more impact when the first exposure occurs in early life, or even when they haven't reported it early in life, when the first exposure occurs in adolescence. And so it does matter when you first experience the abuse. Now the reality is that this is, of course, and this is where we'll get into the tricky nature of understanding environmental impacts, because even though if your first exposure to physical abuse was between 0 and 5, what are the odds that you continued to be exposed to physical abuse all the way through your adolescence? They're pretty high, right? So it's not, that's a different way of looking at the data. But this, I think, is nice, and I could have done this with sexual abuse, and I could have done this with emotional abuse, and you would have seen the same pattern across these 3 variables, which is to say that if you have it very early in life, or if you experience it again in adolescence, when we're having, again, more hormonal changes, right, and probably more plasticity in the response of our brains, that it can have more of an impact on your outcome. This is what I spoke to a minute ago, which is even if you have the time to measure, when did you first experience trauma, that still doesn't answer the question of how long did it last, right? So we know that in some cases when people have abuse, it's not a one-time episode. So in this case, we're looking at sexual abuse data. And how long did it last? So if you're living in the house with the person who's sexually abusing you, or if he's some part of your life, a coach, some other Western community, it can go on and on for a period of time, or it can happen only once, and that matters particularly. So here are 2 graphs, and this shows that really, the impact of sexual abuse is dependent on how long it lasts, and it's particularly dependent on how long it lasts when it first happened in adolescence, right? So here, or not when it first, but when it happens in adolescence. So this is abuse that occurred at 13 or less, and this is abuse that occurred from age 14 to 17. Now, some of these people are overlapping, meaning that their abuse occurred here and continued right on here. But some of them is not. And so we see a particularly strong effect. So when people experienced during adolescence, from essentially age 13 to age 17, sexual abuse that was ongoing, we see that 81% of them end up with post-traumatic stress disorder. So it's a very high rate. It's not surprising, really. It's almost a better question maybe, what about that other 19%? How did they not develop PTSD? And it may be that they didn't, or it may be that they're just 1 point below wherever we used our cutoff for post-traumatic stress disorder. So it's likely that it has an impact. But this is showing that when we look at environmental factors, we also have to ask about when did it happen, and how long did it last? Right, let me take a deep breath here. So what have we said about all of this? So if we step back and say, what are our challenges? So we want to keep doing psychiatric gene by environment research, but where are we now, right? The first is that relatively few candidate genes have been examined. That's because there's a lot of genes and only so many we can either look at or can afford to look at in any given lab, right? Environmental factors in general in the research are generally only coded as present or absent. That doesn't leave us with very many models for assessing cumulative environmental exposure levels or other key factors such as duration or age at time of trauma exposure, which we know from yesterday is so key. Then we have tremendous, so that leads to tremendous variability in what is the data that's going into the models we're examining. And then, you could have someone who appears in one person's study as abused and in another person's study as abused, but they could have had very different experiences. And then variability in the definitions of environment. So often individual level environmental factors are considered, but we don't really look at broader environment. So I haven't spent a lot of time talking about it here, but we could talk about the neighborhood, right? So we've done some data now looking at census tract data showing that the neighborhood that you live in and the rate of crime in your neighborhood interacts with all of these other individual level factors that we've talked about to change your outcome. And then we spend a whole lot of time, by nature of our research, and this will come to this afternoon, is focusing on risk-conferring environments. So that's our focus here, abuse and other things. But we haven't spent nearly as much time focusing on, what aspects of your environment, if present, mitigate risk. Because we know that even despite, if you look at the numbers and types of trauma I present, the rates of PTSD actually look relatively low. So what are the factors going on that are mitigating the risk, right? Because that's at least as important in a process, but tends not to be where our eye is. So we need to look more at characteristics, and then lastly, characteristics of the population being studied. And this is an example of gender, because in many of our genetic studies and most of our GWAS studies, where we look across the genome, we're combining men and women in our samples. But we know from other research that the biological effects are not the same. So these are two different studies we've done at Grady Trauma. One that's published looking at a PACAP gene, which is a stress-related response gene, and we found a genome-wide effect in women, but not in men, right? And this is a soon-to-be-published finding where we looked at genome-wide data on emotional dysregulation, and we found an impact for the IL-2 gene. But we found it in men and not in women. It was sort of flatlined in women. And so we pretty much regularly now look at our data dividing by gender, because when you're thinking about the biological downstream of these things, not to take a look at gender doesn't make a lot of sense. And then this is the gene bisocial environment that we talked about earlier, that same gene that we talked about just a minute ago, very quickly, PACAP. Here what they're showing is that its impact, the number of risk alleles you have for that gene, varies by how high or low the crime of the neighborhood you live in is. And this data comes from the Detroit data. So we often use the Detroit study because it's probably the study which has data most comparable to what we're looking at in Atlanta. All right, so then I also want to talk a little bit about positive family environment so that I don't end on such a negative note, right? We haven't only measured abuse and trauma in our sample. We've also measured both positive family environment during childhood, and we've measured positive environment. Not presenting it today, but we've looked at community involvement for children and other in school and sports and other things. And what we found here is that childhood maltreatment, even after you control for childhood maltreatment and PTSD, if you're looking at the impact, and this is looking at positive affect, so good mood, right? Its impact, positive family environment trumps those things. And that's also true when you look at it with respect, I promised I would speak just briefly, but I have about 30 seconds, about an oxytocin gene. This is a gene that's been shown to be responsive to both positive and negative aspects of the environment in a way that reflects not, because you think about it, it doesn't make sense to ask the question, did we have genes that made us only respond to stress? What we probably have is genetic variations that make us respond to our environment, good and bad, and make us susceptible to the environment. And this is a gene where research has suggested that it's responsive to both positive and negative environment. And when we look at that in our Grady sample, what we see is that it interacts with family environment to predict the positive affect. And depending on which, so this is a place where we sort of close out with that same sort of different effect. We asked people whether they were in warm or stable childhood environments. We put some variables together. And if their home environment was neither warm nor stable, then depending on which genotype they had here, there was a significant difference in positive affect, right? But if they had warm or stability or both, this gene here, So this is the gene that's reflecting sensitivity to environment, the genotype, right? In the absence of either warmth or stability, they show very low positive affect. But in the presence of warmth, stability, or both, they show stronger positive affect, suggesting that the folks in this group are responding to their environment, both for good and for bad. And so the genes may in fact be reflecting sensitivity to environment itself, which is not my idea, but the idea of people much smarter than me, but another way of thinking about gene by environment interaction data. And I think that brings me in almost on time.
Great. We have time for a question or two.
Hey.
Yes.
Did I go first? Okay.
Oh, I'm sorry.
Sorry,
Go ahead.
it's fine. Thank you for that talk. That's a lot of very harrowing data, I thought. So given that these environments are constantly re-exposing people to trauma, for clinicians and healthcare workers, is there anything you can do with individuals, either therapy or pharmaceutically, to treat these traumatized populations, given that they're continuously re-experiencing these traumatic events that could, in theory, sort of undo any benefit of therapy? Or do you have to focus on changing the traumatic environments themselves?
Well, I certainly hope that there's something we can do therapeutically, both with psychotherapy and medications, because I think that that's a place where in our professions we have a shot. That being able to change the re-exposure in the environments is a much broader socio-political question that I don't know, have even less of an idea about how to respond to it. My answer to that is yes, because as we saw yesterday with talking about interventions for kids who've been exposed to trauma, that if you teach them to understand what is trauma and what is a response to trauma, what is a cue, then they can apply that across new things and experiences in their environment. So if we can give them the abilities to sort of put words to and understand what's happening to them, then it can help mitigate the response to the next thing and the thing after that, right? And the same thing's true for parents raising kids in these environments. So along the way, I mean, these are people who've survived a tremendous amount, and I guess as many of them along the way have learned a lot of skills for adaptation. Some of them we may be able to help. And the same thing's true with biological response. If we can help with medications so that you're responding better, so you're less depressed, so that your other parts, so that you're just, then it's going to help the next time a stressor comes along. So I mean, you could look at either way. You could either be drowning no matter what you do, or you can say that at least you have a chance to swim upstream, right, and try to at least have it be better.
George, do you have a question?
Sorry, I asked too many questions. So this, first of all, I want to preface this, but I think this is brilliant, the way you're looking at the childhood and the longer term impact of these interactions is really clever and interesting. So what I'm gonna, I don't want the question I raised to sound like a criticism.
It's okay, you can criticize me too.
Well, it's not a criticism, but it's a concern I have, and it's a kind of a general concern, though, with this kind of research, and I think it's a very difficult problem, but the concern is having adults who are not doing well or struggling with psychopathology describe their past.
Yes.
And there's a lot of potential for memory bias. We know from memory reconsolidation research, which is very impressive, that memory is a biological phenomenon, that we can change the past, we can improve it or we can downgrade it depending on how we re-experience it at the time. So it raises concerns about sort of going, the causality seems to go from childhood to adulthood, but it could be actually going from adulthood back to childhood, that people with suffering from a lot of PTSD may be remembering or exaggerating abuse. And I don't mean inventing it or anything like that. I just mean, yeah.
No, I think that's right. So I think the answer is yes. That I don't think that we can in any way account for all of the abuse or the trauma by a memory bias. I don't think that's the case. But is it true both in our research that we know that probably your genes influence how you remember things, much less how the environment you're in or what you're doing? But I certainly know clinically, you know, I'll talk a lot about, I'll have people I work with in the VA who were exposed to almost the same situation, and one will come away saying, I could have been killed, and it's a bad negative memory for them, and the other one will come away saying, well, thank God I could have been killed, right? And it's a positive memory for them, and so they might not report it as the same. And the second one's generally doing better. So I don't know the right answer. I think we only see this as one slice or one way of looking at the data. Yeah. And then we also need to go back and ask this way, because we know when we do therapy, often what we're asking people to do is go back and change how they look at their past, right? And if we can do that right, then we help them, right? And we don't want to start by telling people who don't think of their past as awful by telling them, oh, well, your past really is awful, you just didn't know it, right? Because that's not always helpful either. So
Okay.
I don't know what to say, but yes, you're right.
That's great. Thank you.
Okay,
Again,
final question.
I work in the area of child neglect, and I study perpetrators, not the children right now. And I wondered, it was so lovely to see an attention to context, because often this work is decontextualized. And the mothers that I see who are from child protection have IQs in the 70s. And often we lop off those folks from our samples, and your sample is much broader. So it's lovely to see the full spectrum. But I do child protection records, so I know how many times these mothers perpetrated. I was interested in how you measure things like assault rate in environment, and then I'd like to comment a little bit about my thinking, because I've collected that kind of data, but in a different kind of way.
But I'm sorry, but assault
How do you
rate?
measure, you said they live in environments with high violence and assault rates and
I mean, there's two different ways we measure it. One is by asking people, right?
Okay.
Either do you see assaults in your neighborhood, what kind? The second one is by, asking them whether, do you think your neighborhood's dangerous? Those have memory bias. The third one is we use census tract data to map their address at the time that they answer our questions. Now that changes a lot, so that may not be right. And our biggest problem in our sample with census tract data is zero variability even with all these census tracts. But that's the different ways.
I had an NICHD grant, and I worked with a geographer here at Penn State, and we're measuring activity patterns. So where do mothers actually live their lives? And the neglect mothers live in almost half the space than
Yes.
the controls. So GIS doesn't really represent
Right.
the neighborhood they actually live in, and trauma may constrain their movement within the
Right, if you
neighborhood.
hardly leave your apartment much, except for maybe to get your kid to school or not, right, then your neighborhood is not as relevant.
So one of the things at Penn State that's lovely is we have methodologists who help us drill deeper into these what seem like simple variables that are much more complex. And the traumatized groups look different in terms of use of their neighborhood. And I think we need more good capturing of those contextual factors and control.
Well, I agree, and I think the signals are that I have to stop talking, but we can talk about this more later. But I appreciate your seeing my research as good capturing complexity. The other way would be messy, so I appreciate your perspective on it.
Well, thank you very much. And there will be an open discussion at the end of the session, so comments about these types of issues I think would make for a good conversation at that point. So without further ado, I would like to introduce my friend and colleague, Dr. Idan Shalev. Dr. Shalev received his PhD at Hebrew University under the supervision of Richard Epstein. He also completed a postdoctoral fellowship at Duke University, where he worked with Avshalom Caspi and Terry Moffat, examining the biological embedding of stress in the Dunedin study. Idan came to us here at Penn State about 2 years ago as an assistant professor in the Department of Biobehavioral Health. He's also a faculty member with the Network on Child Protection and Wellbeing here at Penn State. His research entails an interdisciplinary approach to the identification of mechanisms underlying the biological embedding of stress across the lifespan. Specifically, his research tests the effects of stress from early life on change in telomere length and other biomarkers of aging across the life course, and the consequences of change in telomere length for physical and mental health problems. His work is especially important for advancing our understanding of how child maltreatment increases the risk for such varied health outcomes, including cancer, obesity, psychiatric disorders, and the like, through the mechanism of telomere erosion and cellular aging. So please join me in welcoming Dr. Idan Shalev. I think he was the first network faculty, is that right?
No, I was the third, after you guys. Thank you, Chad. Okay, so this is actually really nice because this session is on, can you hear me? Okay. So this is nice because this is a session on genomics, and Kieran was talking about epigenetics, and Bekh was talking about genetic variation and gene-environment interaction, and I'm going to talk about a different genomic element, which Chad mentioned. This is telomere length. And telomeres are the elements that are found at the tip of the chromosomes, and are considered to be this kind of a clock that regulate how many times a given cell can divide. So this is why you see the chromosome and the clock in the background. And I'm going to present to you two studies that were already published. I'm going to talk briefly about the next steps and the current research that I'm currently doing in my lab. As you know, science takes time, so unfortunately I don't have anything really new to show you. But I'm going to discuss about two studies in relation to early life adversity and telomere regulation. So I want to start first with two important frameworks that are relevant to the studies I'm going to show you. So the first one, Kieran actually mentioned that. This is the developmental origin of health and disease model, also known as the Barker hypothesis. And this is a model that suggests that conditions during pregnancy, which can influence the environment of the fetus, can have long-lasting effects on our health. So in other words, early adverse conditions in utero may have permanent changes on our body's function and metabolism, which can result in accelerated aging later on. And this is an important concept to consider because I'm talking about telomeres as a mechanism for aging. And aging, and this is not a new concept, aging is a lifelong process that begins already right after conception. So we don't need to wait until old age to study aging processes. Okay, so this is the first model. The second one, which is relevant, and I think Bekh actually mentioned that, is the CDC Adverse Childhood Experiences study. And this model proposed a whole life developmental cascade, leading from adverse experiences in early life to social, emotional, and cognitive impairment, then adoption of health risk behaviors, leading to disease and early mortality. Now this is a landmark in epidemiological research, and the Barker hypothesis, the developmental origin of health and disease, is also a very important model, but both of these models actually emphasize some scientific gaps in our knowledge. And specifically, both the DOHAD model and the adverse childhood experiences lack the biological mechanisms. Okay, so if we're thinking about the developmental origin of health and disease, epigenetics is actually considered to be a mechanism that can explain the embedding and the impact on later life health outcomes. But here we're missing some of the biological mechanisms that can explain this embedding, this long-term effect. And this is where I come with the study of telomeres. So telomeres, if you never heard about this before, telomeres are specific genetic elements at the end of all linear chromosomes. They're composed of repetitive DNA sequence, the TTA-GGG, that repeats itself hundreds and thousands of times. And together with other protein complex that are bound to these genetic repetitive DNA repeats, they form this kind of cap end structure at the end of the chromosomes, okay, to protect the chromosome from DNA damage response. And the best way to think about it, there is an analogy when it's very similar to the plastic tips that we have at the end of the shoelace, okay, that protect the shoelace from fraying. So this is a very similar function the telomere play on the end of the chromosomes. Now, a known phenomenon in biology is the fact that telomeres are getting shorter each time the cell is dividing. Okay, this is